Okay, so for this audio lecture, we'll be covering chemotherapy, which is covered in chapter 12. When we're talking about chemotherapy, we're talking about drugs or antimicrobials. Antimicrobials is a general term that talks about drugs that are against microbes. You can use more specific terms like antibiotic. An antibiotic would be specific to a bacteria. Antiviral specific to a virus. Antifungal specific to fungus, fungal, so yeast and molds, and antihemolyth for parasitic worms. So just again, keep in mind that antimicrobial is a general term. So like on an exam or an assessment piece, if I use a specific type of antimicrobial drug or asking for a specific one, then do make sure you're fitting it to the category that's being asked. If I'm asking just about antimicrobials in general, then that's where you can give me a general example. So when we look at antimicrobials and where they originate from, originally they were isolated from different types of microbes, because keep in mind microbes are having relationships with other organisms, and so they need to produce products, metabolic products that can fight, can injure those microbes around them. So examples of this would be streptomyces and bacillus. Those are bacteria that different antibiotics have been isolated from, but also penicillin and cephalosporin. So penicillin is the fungus, is the mold in which penicillin was originally isolated from. Since kind of the discovery of these early antibiotics, what then chemists did was that they would actually synthesize new drugs based on these naturally occurring ones. So they would change side groups, so you would get different classes of these antibodies. Kind of the different side groups that could be used have been utilized, and so this is kind of like going back to the drawing board, where you're looking for new sources of antibiotics from microorganisms, and that's where tiny earth comes in, right? So in tiny earth, you're taking soil isolates, microbes isolated from the soil, and you're looking to see whether they inhibit the growth of other organisms, and you get that ring, that area where you don't get growth around that, and that would indicate that that soil isolate, that microbe that you isolate from the soil, is producing some kind of metabolic product that's inhibiting the growth of your tester strain, which would be one of those safe relatives of the escape pathogens. And so then you would further isolate it, and then get a pure culture, and then you could do antibiotic extracts. You could sequence it to identify what genus or even species it is. So again, the idea of the tiny earth is being part of the group of microbiologists, chemists, who are going back to the drawing board, and kind of looking at it afresh. So with antimicrobials, and more specifically antibiotics, they have several different targets or mechanisms of action. And so this figure very nicely shows you this kind of visual of, we have a bacteria here, and there's different cellular structures, and this is where Unit 1 was really important. So there's different cellular structures in the bacteria that we can actually target. And good targets for antimicrobials, for antibiotics, are going to target what's different, right? So we want to make sure that it has low toxicity to us, the humans, the host, or if you're talking about veterinary medicine, whatever the host is there. And so when there's something different between them and us, it becomes a nice target. And so bacteria have cell walls. We don't. So we actually see a large group of antibiotics that are cell wall inhibitors, so they can either block the synthesis or the repair process. So keep in mind that in order for a bacteria cell to divide, it needs to have enough cell wall material to support two cells. Also, there's just normal repair that has to happen. Peptidoglycan gets damaged, you have to put new peptidoglycan in. And so antibiotics like penicillin, cephalosporins, vancomycin, they can inhibit the ability for the bacteria to be able to either synthesize or repair. Then when we look at the cell membrane, we can disrupt the cell membrane being selectively permeable, and so detergent-like antibiotics are used. So polymyxin would be an example of a cell membrane disruptor. So looking at nucleic acid synthesis, that's also required for the cell to divide, but also to make proteins. And so we can affect the ability for the bacteria to be able to produce nucleic acids and put them together to form DNA or to form RNA. So inhibition of replication and also transcription being the DNA to RNA. And so we can affect the enzymes. So the polymerases that play a role, or helicase, gyrase, so all these other enzymes that are involved in those processes. And so the quinocones are a common antimicrobial that targets this process. One of the things to keep in mind is that the process of replication and transcription is kind of conserved between prokaryotes and eukaryotes, and so there could be more toxicity with these. Some of these enzymes are more specific, so there's differences between our eukaryotic enzyme versus the prokaryotic enzyme, so then those become better targets to go after for potential antibiotics. Another large category besides cell wall inhibitors is protein synthesis. So keeping in mind that their ribosomes are 70S and our ribosomes are 80S. So again, it's different in them versus us, so we can target that. When we look at protein synthesis, keep in mind that the ribosome is made up of different subunits, and so we can target we can have antibiotics that target a specific subunit or both of the subunits. And so I don't expect you to know which subunit it affects, but you can see how there are antibiotics like clindamycin, erythromycin that are going to target this 50S subunit, and then we have the aminoglycosides like gentamicin, streptomycin, but also tetracycline, which an example would be like doxycycline, that would target the 30S. Okay. And then lastly, we have the category of folic acid synthesis. So some textbooks call it vitamin synthesis, but the only vitamin synthesis I've ever seen targeted is folic acid. So just beware, it's probably the only one you're going to see. And so we can have antibiotics that target folic acid synthesis because we as humans don't actually produce folic acid. We lack the enzymes to do that. So we actually have to get folic acid from our diet, where microbes actually produce their own folic acid. And so they have the enzyme systems to do that. And so we can have antibiotics like the sulfa drugs that will actually target those pathways so that the bacteria are not able to produce folic acid. And if they can't produce folic acid, they can't make DNA, RNA, and they can't make proteins, and so the cells will end up dying. So just in case you didn't like the figure and you'd like it listed out, here's the different mechanisms of action. So we'll look at each one of these a little more specifically. So with cell wall inhibitors, again, we can be inhibiting the synthesis or the repair process, and so there's going to be constantly this demand to synthesize newly polypeptidoglycan, putting these subunits together to create the cell wall. And if you don't, then you're going to have these weak points, and then that cell could end up lysing. So again, penicillins and cephalosporins are two groups of antibiotics that fall in that category. Within the penicillins, you would have amoxicillin, ampicillin, methicillin, and then with the cephalosporins, there's a number of generations with the cephalosporins also. For these antibiotics, they're known as being bactericidal. So if we look at that word and break it apart, bacteria, and cidal kill. Okay, and so just be aware that with antimicrobials, they can be cidal where they actually kill the microbes, or they could be static, and that's where they don't kill the microbes. So it's not like they're lysing them and destroying them. What happens is that they can't support cell division, so they can't divide, and so you keep the same number of microbes present. This is a concept we'll talk about more with the physical control and the different methods, but just again, be aware that some of our antimicrobials will be cidal, and with cell wall synthesis inhibitors, they would be an example of the bacterial cidal. So as you can see in this figure, we have up at the top, we have this normal bacteria cell. It's got peptidoglycan on the cell wall, and now this bacteria gets exposed to a cephalosporin or penicillin. So, its host has now ingested this antibiotic. And so what happens is that it's not able to repair the cell wall. And so you can see how they're just getting these weak points. So the membrane is going to actually bulge out there, and then the cell ends up lysing. And just so you know, just as a reminder, when we're talking about the cell wall, we're talking about all of these different subunits of peptidoglycan. So the NAG and NAM that you see, each one of those are peptidoglycan, and they're cross-linked together. And so if one of these becomes damaged, it becomes a weak point within that. So then looking at nucleic acid synthesis, keep in mind that there's several different mechanisms that can actually be blocked because these are multi-step processes. So think back to an intro bio class when you would have talked about DNA replication, or you would have talked about transcription being that DNA to RNA process. And so these processes are multi-step, right? You have to memorize all the different steps, the enzymes coming in to open up the DNA, to then add the nucleic acid, and move along, right? And with transcription, translating that into RNA form. And that we need each of these nucleotides to be present. So if you need A has to be there. If you need a T has to be there. And so we can block the synthesis of the nucleotide, and so then there's no T's available, so you wouldn't be able to add that, so you wouldn't be able to make DNA, or you wouldn't be able to make RNA. And again, we can also affect the enzymes that are involved in the process, so we can inhibit replication there and opening of the replication fork. That can't happen. So again, the overall process of DNA synthesis or the production of RNA would be blocked. With protein synthesis, what we're doing is we're going to inhibit the interactions between the ribosome and the RNA complex from forming. And again, because the ribosome is a different size than ours, there tends to be a lower toxicity on these drugs, just like with the cell wall inhibitors. And so again, these become our kind of go-to, more preferred antibiotics because of that. Now, just like with DNA synthesis and replication, these were multi-step processes that you would have learned in intro bio. So was protein synthesis. So you probably talked about protein synthesis in an intro bio course, and you would have talked about how these subunits for the ribosome come together, they bind to the RNA, and then they're moving down the RNA. And while they do that, you have your transfer RNA present. It brings an amino acid in, shifts down to the next codon. You have another transfer RNA coming in with amino acid. You get a peptide bond, and then you continue this translocation down the RNA in order to make your protein, right? So all these steps are happening, and they're the different drugs, the different classes of antibiotics would affect different components of that. And so a common antibiotic would be tetracycline, so that's second from the bottom. And so it's actually going to inhibit the transfer RNA from coming into its site, so that you can't or it blocks it so it can't come in, so it means you can't get the amino acid being added, okay? With erythromycin, that one's right at the bottom, that one actually prevents the ribosome from moving, so it kind of locks the ribosome in so you can't move to the next codon to get elongation of your amino acid. And again, there's additional ones. Another one that's common is the aminoglycoside. And so what it does, it actually causes a misreading of the RNA so that you don't get the correct reading, so you don't get correct production of your protein because it gets misread. So again, I don't expect you to memorize like which, I'm not going to be like tetracycline, how does it work? What does, but again, it's of interest. And again, it's one of the reasons why it's a large category also, because you can have drugs that are affecting different parts of the pathway. With cell membrane function, we want to keep in mind that our cells are made up of cell membranes, and there's a lot of homology between prokaryotic cell membranes and eukaryotic cell membranes. And so the idea is that you're going to target lipids that are unique to microbes they're in their cell membranes. And so it is a more limited category, just because, again, there's the risk of toxicity. So most of these like polymyxin, tend to have like a detergent type look. So they almost look like phospholipids, because that's what detergents end up looking like, so that they can get incorporated into the cell membrane and then they disrupt. Your antifungals tend to have the same approach as they incorporate into the cell membrane, just so that way. Antifungals don't tend to actually target the cell wall. They tend to target the cell membrane, and in that case, some of the antifungals tend to have more toxicity, just because, again, our cell membranes are very similar to fungal cell membranes. And then the last category, again, would be either vitamin synthesis or folic acid synthesis, is the more specific, and I would say the more preferred. And so the way these work is by competitively inhibiting. So the sulfa drugs actually compete for binding sites on enzymes. And so because you're doing competitive inhibition, you need to give this at a high concentration. This is a concept you would have talked about in like a chemistry class. And so you can see here on this figure how we have PABA. This is the substrate that's going to bind to the enzyme. And what happens to PABA is that it gets eventually converted into folic acid. So just keep in mind it is a multi-step process, and so that's to our benefit because there's multiple enzymes involved. So each arrow you see in that pathway is an enzyme that is involved, and so we can have drugs that target those enzymes and compete for those binding sites. So again, you have PABA that's going to bind to the binding site on the enzyme, and if we give sulfa drugs at a low concentration, we can still get this interaction with the enzyme and the substrate, which means folic acid would still be produced. But if we give it at a high concentration, which we see in the last panel there on the right, what happens is it competes for that site so that PABA isn't able to bind because there's too much sulfa drug there binding and blocking the ability for your substrate to bind. And so in that case, that organism wouldn't be able to produce folic acid, and which means that they can't produce amino acids, so they can't make proteins, and they can't make nucleic acids, so they can't make DNA and RNA. Okay. So a concept related to antibiotics and more so antibiotics, but antimicrobials too, is the idea of spectrum of activity. And so with antibiotics, we can have something that has a narrow spectrum of activity. So for a narrow spectrum activity, that would only work against a single type of bacteria. So if you look in this chart, you can see how with polymyxin polymyxin works best against gram-negatives, and it doesn't have as much of an effect with gram-positive or chlamydia or rickettsias or microbacteria. So again, it's really only good with the very specific types of bacteria, so it would be considered narrow spectrum. And that's because the gram-negative bacteria also have the outer membrane, so you would disrupt the outer membrane and then also the cell membrane. Penicillin is also generally considered narrow spectrum. So they work as cell wall inhibitors, and if we think about which type of bacteria has a thick cell wall, that would be gram-positive. You'll notice that on this that there is some cross-reactivity with some gram-negative bacteria and some of the chlamydias, and that's generally some of the newer generation of antibiotics in which the side chains have been changed, and so there's some cross-reactivity, but we don't quite understand why. But for our case and for simplicity, we would say that penicillin is narrow spectrum. So I will mention this in class, but I'm going to say I do not agree with WebMD, okay? And this is where I write the exam, I grade the exam. So for our purposes, penicillin is narrow spectrum. And that's, again, because if we think about the mechanism of action of penicillin, it's to inhibit cell walls. Which bacteria has the thickest cell wall? Gram-positives, right? Gram-negatives wouldn't tend to have reactivity to penicillin because they have an outer membrane, so we're not going to get the antibiotic in as effectively, and then they only have a very thin cell wall, and so it's not going to be very effective. And so why it works on some gram-negatives, again, we don't really know. We don't understand clinically. Sometimes we just don't care. And so whether some other mechanism is at action, whether there's some cross-reactivity to something else, we don't know. But again, for our purposes, we are going to consider it narrow spectrum. Now, an example of broad spectrum would be tetracycline. So with tetracycline, you can see how it covers gram-negative, gram-positive, chlamydias, and rickettsias. So it works against a number of organisms. And so with tetracyclines, again, broad spectrum, because broad means it works against multiple types, multiple categories of organisms. Okay, and so when you have a broad spectrum antibiotic, it's really good for if you don't know what's causing the infection. So if you have a patient coming in and you're like, we want to start treating them, but we haven't gotten the lab results back yet, that's where you would give a broad spectrum, because then at least you're starting antibiotics, at least you're starting to treat the individual, opposing to waiting, 2-3 days before lab results are coming back. Okay, so again, you should be aware of the concept of narrow spectrum versus broad spectrum, and you should be able to give an example of each. Now, when we look at mechanisms of resistance, obviously antibiotic resistance is becoming an increased concern, hence why with the Tiny Earth Project, we're being part of this group that is looking for new antibiotics. And there's many labs that are using other approaches also to develop new antibiotics. And so again, resistance is a great concern, and we'll talk more about resistance in a moment, but how it arises, there's several different mechanisms. So the first mechanism is we can actually have an antibiotic that will actually have an enzyme that the microbe has that actually is able to deactivate the antibiotic. So you can see how we have penicillin present here, but a bacteria could produce penicillin ACE. ACE meaning enzyme, penicillin is its target. And what it does is it breaks down the penicillin so it can't have an effect in the cell. Okay. Another mechanism a bacteria could have is that they have decreased permeability. And so this is where we can actually have changing of the receptor So there can be some kind of mutation to the receptor that spontaneously happens. Now it gets selected for. So the drug before could bind to the receptor, but now the drug can't. And so if the antibiotic requires the receptor in order to enter into the cell to have an effect, then it wouldn't be able to enter the cell. Microbes can acquire drug pumps, so they can pick up genes in order to have these drug pumps, these proteins, across the membranes. So the drug comes in through its receptor, through its mode that it enters the cell, but then there's this drug pump that will actively pump the drug out. So it's kind of like, drug gets in, the drug then gets kicked back out. Drug comes in, drug gets kicked back out. And similar to the receptor, where if the drug's not inside the cell, it doesn't function. So think about if you have a drug that affects protein synthesis, it needs to get in the cell to bind to the ribosome, to bind to the messenger RNA. And so if it can't get in, then it can't have function, right? So again, with the drug pump, you're just constantly pumping the drug out. So the bacteria is able to survive because whatever process the antibiotic was going after cannot be inhibited. Another mechanism is where the binding site gets changed. So this could either be, there's been a sequence change in the ribosome or a sequence change in the enzyme it binds that's going to inhibit replication or transcription. So maybe the RNA polymerase has a mutation on it, and so the drug is no longer able to bind to it. So thus it's not effective. The other mechanism is that it can have an altered metabolic pathway. So normally you go from a substrate to product, and maybe this goes through several steps, but the drug acts on enzyme, B, and so it will actually go around that and create a new pathway in order to produce the product, okay? And so this is definitely with viruses, but also with bacteria. This is an area where we see resistance, where they create these alternative metabolic pathways. So we want to keep in mind with antibiotic resistance, it happens when there is selective pressure. So the reality is, if we were to think of ourselves as a petri dish, we are a mosaic of different types of microbes. Some of the microbes have resistance and some of them don't, right? So this plate shows you where we have these microbes that do not have the drug resistant, where we have some that do. Once we put a selective pressure on them, and because most of our, all our antibiotics we're using currently work on killing or stopping growth, halting growth, what we're going to end up with is what we're seeing in the middle one, where early on some of the microbes are going to be killed, but by the time, it's late, we've killed all the microbes that are sensitive to the drug, which means only the resistant ones are left, which means they're now present and able to repopulate. And it's only the resistant ones, right? Because all the sensitive ones, all the ones that would die or stop growing in the presence of the antibiotic are gone, or they're halted, and so the only ones that are able to grow and actively divide and then repopulate would be the ones that are resistant. So this is one of the reasons why antibiotic resistance is of concern, because now you're starting with a population that is resistant, and if you need the drug, that all the ones that are present already had that mutation that you've now selected for. So how do we prevent drug resistance? One's to limit drug use. So the less drug use that's happening, antibiotic use, antimicrobial use, it means that there's less of this selective pressure, and so really we're all continuing to walk around like that first plate where we're this mosaic. And so, yes, you might still have the resistant ones there, but we're not actually selecting for them, right? And the more of us that are mosaics, the better off. The thing to keep in mind, if you think about where antibiotics are being used, most people think it's actually in human medicine, and the case is that most antibiotic use is actually in veterinary medicine, and that includes agricultural. So agricultural use is kind of lumped into veterinary medicine. And so when we look at the use of antimicrobials, 80% is used in veterinary medicine. And again, that includes agricultural purposes, where less than 20% is used in human medicine. So yes, it's really important in human medicine that we're mindful of antibiotic use and try to limit its use to just when it's needed. But the reality is we also need to look at how we're like livestock and domesticated animals, and how are we treating them, and are we also being mindful of antibiotic use in them. And so the USDA has come up with guidelines as far as removal of antibiotics from food. So it used to be that many of the foods which livestock were consuming would contain antibiotics. Farmers would also use antibiotics to prevent illnesses, which really isn't that great of a strategy. And also, the North American Veterinary Medical Association and other veterinarian associations have come down with guidelines as far as use of antibiotics. Antibiotics that you used to be able to just go to your feed store, or, your Tractor Supply, and purchase. You can no longer purchase. You need to have a prescription in order to purchase those. So, definitely in that agricultural veterinarian group, there's been that they are aware that they're part of the problem. And so again, it's not just changing human medicine. We also need to look at other areas that are utilizing antimicrobials. We also want to make sure that we have proper drug use. So if you have a cold, if you have a virus, an antibiotic is not going to be effective. Just like if you have a bacterial infection, an antiviral wouldn't be effective. And so making sure that you're using the correct drug so that you're actually eliminating the pathogens that are actually present. And with that, also making sure that you're using the full dose. And so if you start to feel better and you stop taking the rest, because then you can save the pills for later on, that's not a good strategy, because what can happen is you can actually have some of these intermediate ones that aren't quite resistant, but they're not quite sensitive. Those ones actually get selected for, and they're able to populate you. And they potentially could also be sharing plasmids with each other. And again, you should always take the full dose so that you don't risk having a reoccurrence or secondary infection. So, there's a reason why you're supposed to take some of them for 7 days or 10 days and some of them for 14 to 21 days, because the mechanism that they work. So cell wall inhibitors tend to have a shorter dosage time, where something like a protein synthesis inhibitor like tetracycline, those tend to be longer course. So those tend to be the 14 to 21 days that you course of antibiotics in order to get effective killing, just because the mechanism's different, right? So a cell wall inhibitor is bactericidal, where with protein synthesis inhibitors, those are bacteriostatic, so they don't kill the bacteria. But if you take them long enough and the bacteria can't make proteins long enough, the bacteria actually does end up dying, but it's not a direct killing. Okay? Another way to prevent drug resistance is by using narrow range antibiotics. So broad spectrum will kill all these different types of bacteria, but only one type is like the cause of the illness. And so you've killed all these beneficial bacteria, right? With narrow range, if you pick something that is specific to the pathogen that you are trying to kill, then you're less likely to have complications. You're only killing the ones in those categories. And so you have all these other beneficial bacteria left behind. Another strategy is by using multi-drug treatments. This is really important if you have one that's developing different pathways, and or even this, if you have enzymes being produced that deactivate. So we definitely see a growing number of infections which multi-drug treatments, a cocktail of inhibitors are being used to circumvent some of these drug resistance issues. One drug that utilized is augmentin. Augmentin is a combination of ampicillin and an acid, and that acid actually deactivates the penicillinase so that the amoxicillin is now effective. So amoxicillin resistance is definitely pretty common, and so you're much better to get augmentin, which is this has 2 drugs in it. And again, one is to deactivate the penicillinase so that the amoxicillin is effective. Augmentin does have some side effects to it, so there is a low compliancy because of the side effects. The side effects are like explosive diarrhea. So I chuckle, but obviously if you end up having that side effect, you tend to stop taking the drug because it's pretty severe. How do we look at drug sensitivity? How do we look to see whether an antibiotic is effective? The way to do that is through Kirby-Bauer, is the standardized procedure. So you would have looked at Kirby-Bauer in the lab. So what this does is uses discs that have different antibiotics on it, and they can be at different concentrations even. You put the disc down on a plate that has the bacteria that's been inoculated all over the plate. And then you look for these zone of inhibitions, and so you can see these circles that are around the disc. The larger the zone generally means it's more susceptible, where a smaller zone means it's more resistant, right? Because smaller zone means the bacteria is able to grow and it's not being inhibited or killed by the antibiotics on the disc. Now keep in mind that some antibiotics, they don't have the mechanism action that's correct for that bacteria, right? They don't have the spectrum of activity to kill that bacteria. And so we just want to be mindful that sometimes no or small zone of inhibition doesn't mean we have resistance. It just means that that antibiotic doesn't work for that bacteria. Okay. And so Kirby-Bauer's can be done to assess what antibiotic would be best for an infection. So if you have a patient who has a known drug allergy and has an infection, you could culture the bacteria and do a Kirby-Bauer to determine which antibiotic to give to the patient. You could also, this would also be utilized for someone who's maybe failed, first round of antibiotic. They would, they could test to find out what would be the next best antibiotic. There's usually, if you have an inkling of what the infection is, there's usually a go-to antibiotic for that infection. There's usually even what's the next one to go to if that one doesn't work. But again, Kirby-Bauers are very useful so that you would know the sensitivity, and especially if you have an infection that is showing resistance, or if you have a allergy to an antibiotic. So with that, we want to keep in mind there's a difference between allergies and sensitivity. So for an allergy, this is where we're having an immune response. So either a T-cell driven response, so usually you would have like a hives or a rash, or an antibody response. Okay, so again, allergy, rash, anaphylaxis usually are the classic kind of signs of an allergic reaction. This is different than sensitivity. For sensitivity, this could be how you process the antibiotic. So we all have different mitochondria, and within the mitochondria, our ability to process different drugs is altered, and so we may process the drug more effectively and make it inactivated, and so we're not going to be as sensitive to it, where other individuals may not process the drug and deactivate it as quickly. And so in that case, they would have more active drug floating around, and so they'd be more prone to toxicity issues, sensitivities, side effects, right? So with sensitivities, a lot of times those would be the common side effects for the drug. So, with that, if you're taking an antibiotic and you're affecting your normal flora, your normal microbiota, you might have some side effects like GI issues, right? Because you're affecting the microbes in your gut. So it wouldn't be surprising that they would be affected, and thus you would have some sensitivity issues or GI distress. Sensitivity to light, where you're more prone to sunburns, is another common side effect or sign of sensitivity. When we look at treatment strategies, this goes into the idea of the narrow versus broad spectrum. So ideally, you always would want the patient to go onto a narrow spectrum, but sometimes you don't know what they have to begin with. And so again, you would use a broad spectrum antibiotic until you ended up getting more information. Sometimes using epidemiology, using what's going on, playing a little bit of detective if everyone's at the same school and they're all coming in with sore throats. Well, right now they wouldn't be able to go to school. But normally, maybe other kids at the school or in their grade have strep throat. And so you would kind of go with the idea that they have strep throat and treat them for strep throat, wait for the culture to confirm, right? You're seeing a little bit less of that because of, there's fast tests that they can do. And so within, 5-10 minutes, they can have a result of like whether you are strep positive or not. And then also culture results are generally, you can get that back in a day. And so, again, a lot of times narrow spectrum would be utilized. Now, if you have a patient coming in and you're not sure what they have, again, you might prescribe a broad spectrum. And then once you have the lab results back, you would call them back and you would say, okay, we need to change your antibiotic. And this is annoying to patients. And maybe this has happened to you. Yes, it would be annoying. But again, it's always best to switch to a narrow spectrum as soon as possible. So next, we're just going to look at some of the different classes of antibiotics. So again, with the different classes, they would have different mechanisms of action, or chemically they're different. They were either isolated from a different organism for their original generation, and then maybe, and that's what kind of puts them into the different classes. So the first class we'll look at is the penicillins. So these were discovered by Fleming. So these are some of our early antibiotics. These and the sulfa drugs would be the earliest antibiotics. And so you can see how the different penicillins, their side group, their R group is different. And so again, what chemists have done is they've just changed out the R group, and they find that there's low toxicity and it still has an effect. And so then those are utilized. So they're all a little bit different than the penicillins. And ampicillin and amoxicillin. So they all have this beta-lactam ring. So you can see that beta-lactam ring. And then penicillinases, also known as beta-lactamases, are the enzymes that deactivate penicillin. So what they do is they break that beta-lactam ring, and so that the antibiotic doesn't function. So cephalosporins have the same kind of structure, that they have that beta-lactam ring, but they have two side groups. So on both sides of that beta-lactam ring, they're going to have these side groups hanging off of them, the R group 1 and R group 2. This means that, chemists can come in and change both of these so that we can have additional different generations. Currently, we're on about generation 8 of cephalosporins, and so again, just changing these side groups gives us a new generation, and it gives us, the earlier generations have resistance, and so now we can utilize those. Now with cephalosporins, definitely in the later generations, we see activity with the gram-negative bacteria. It's still believed that the mechanism is where we block the cross-linking of peptidoglycan. There could be some other kind of activity that's allowing it to work against gram-negatives. But again, in medicine, sometimes it's not so important about the details. It's just it works. And so some of the later generations of the cephalosporins are prescribed for gram-negative infections because they do help in clearing the infection. With aminoglycosides like streptomycin and neomycin, which is neomycin's the generic for Neosporin, it's going to bind to the 30S ribosome, and so it changes the shape of the ribosome so the subunits can't come together, so it causes translational errors. With tetracyclines, they're going to block the A site, the active site, so that the transfer RNA can't enter. So the important thing to keep in mind is that this is reversible, and so if you stop taking your antibiotic early, the transfer RNA will start to move into the A site, and then the bacteria can start to make proteins, and then the bacteria wakes back up and causes an infection. Because with tetracyclines, they're bacterial static, again, so it kind of like freezes the bacteria, not temperature freezes, but it's kind of like, you're playing freeze tag and you freeze and you can't move. So with tetracycline, the bacteria can't make proteins, and so when you can't make proteins, you can't function as well, you can't grow and divide. So again, it doesn't kill the bacteria outright, it just kind of makes it so it can't grow and divide, and so you can't increase the numbers. If the tetracycline is around long enough and it can't make proteins long enough, then in that case, the bacteria would die. And so this is why long courses of treatment are needed for tetracycline. So with tetracyclines, so one other interesting thing about tetracyclines is tetracyclines affect your ability to absorb calcium. So tetracyclines are not recommended for young individuals, individuals who are still growing bones. So the FDA says anyone under the age of 14 should not receive tetracyclines, doxycycline. Doxycycline is common treatment for Lyme's disease because originally we didn't know what caused it. But penicillin can be used, and so for young children, usually penicillin is used. Again, the FDA says 14, but keep in mind that growth plates aren't fused, and so it's actually best to wait until growth plates are fused, and so it's more like 18. That most doctors will follow that guideline, and I would advocate following that guideline of just being cautious to make sure that you're not affecting calcium absorption. And then Chlorophenical binds to the 50S ribosome subunit. So it actually prevents peptide bond formation. There is high toxicity to this, so it's known to cause a condition called aplastic anemias, and this is also referred to as gray baby syndrome. So what happens is that for some reason the drug will actually cause the red blood cells to lyse, hence anemia. And so if it happens in young kids, they'll end up going from this really nice pink color, and because the red blood cells are lysing, they will then go to this gray color, and if it's not caught immediately and blood transfusions given, it can be lethal. And so it is used only in very rare cases for young children. So there are cases where with premature babies, if they develop infections and other antibiotics aren't working, that they would utilize this. Usually it's more common used in like eye drops. So for bacterial infections of the eye, because it's a lower dosage and you're not going to get as much absorption into the blood system through the eye. And again, it's given in extremely low doses if it's used in eye drops, where if it's used for like a preterm baby that has an infection, the dosage has to be a little bit higher, and then again, you're more likely to get these aplastic anemias, which are still very rare, but obviously if they happen, because they are potentially lethal, they're, medically, they're going to try to avoid that situation. And they would try to utilize other drugs. Oxyzonone will bind to the ribosome, and so it actually prevents the initiation. Linezolid is one of the common antibiotics within this group. And as we mentioned about sulfa drugs, they are going to be given at high doses so they can competitively inhibit. And again, just like with penicillin, cephalosporins, or some of the older antibiotics, we don't tend to see the resistance in the sulfa drugs. And part of that, I think we kind of we went away from using sulfa drugs. So sulfa drugs have kind of become more popular, and I would say more popular in veterinary medicine than necessarily in human medicine. There is cross-reactivity between sulfa drugs for bacterial infections, but also for fungal infections too. And then for antifungal drugs, as I mentioned, many of these have kind of a detergent look to them. So you can see here how you've got those kind of like those phospholipid tails. So they're able to bind into the membrane and disrupt the membrane. Because funguses are eukaryotic and we are eukaryotic, we do have to keep in mind that some of those antibacterial drugs aren't going to be effective against fungal infections, and some of the antifungal drugs are going to have more toxicity to us as humans than we would see with like antibacterial drugs. So with antiviral drugs, there's going to be 3 modes of action. With viruses, we have the entry of the virus into the host cell, and then the replication, where we're going to utilize the host cell's machinery in order to make the viral molecules that we need to make new viruses. And then we need those molecules to come together to assemble so that they can be released. And so that's the gist of how to make a virus, right? And so antivirals can inhibit or block one of these mechanisms. And so they're put into 3 different mechanisms of action or modes of action. And so one is to prevent the entry into the host cell. And so this is by blocking receptors that are on the surface that allow the entry. The next mode of action is where we block transcription and translation, so the making of these viral molecules. And then lastly, preventing the maturation of these viral particles. That's the assembly and the release, okay? And so this chart does a nice job of summarizing the different modes of action. So again, entry, production, release, and I would say assembly and release. And it gives you some different examples. And so like Tamiflu is going to block the entry, hence why it's really important to make sure if you're going to take Tamiflu that you take it early on, because it's not, once it enters into the cell, Tamiflu is not working on these other modes of action. It's trying to block entry before it has an opportunity to enter the cell and replicate. If we look at acyclovir, that would be a inhibiting nucleic acid synthesis, so that replication and production of the different viral particles. And then cyclovir, that's where we would have the inhibition of assembly and release. Okay, so normally I don't go too much into the antivirals, but I think what we'll do is we'll actually look at some of the different antivirals as, you know, one of our class activities, because I think right now it's a pretty hot topic. So some of the drugs that are looking as potential treatments with COVID are antibodies that actually target the immune response that is activated during COVID and other viral infections. And so later on when we talk about the immune system, we'll talk about complement. And so there is a protein involved in the complement pathway that's known as C5a. And so this actually causes the cytokine storm. And so there's an antibody that actually targets and binds the receptor for that, so that the idea is that you wouldn't get as many cytokines being produced and you don't get all that damage that actually is caused. So in some cases, some of these viral treatments aren't looking at this blocking of replication. So again, we'll look at this more so in a class activity. So that's going to conclude our audio lecture for Chapter 12, Chemotherapies, Antimicrobials. And as always, if you have questions, please contact me.